Assalamu alaikum class. Today's lecture is about cells, tissues and organs of immune system. By the end of this lecture, you will be able to understand the different types of cells and organs of immune system, how these cells originate from different lineages, and what are the structure and function of these individual cells, and what role the lymphatic system, its organs and tissue plays in generating an immune response. The immune system consists of many structurally and functionally diverse organs and tissues that are widely dispersed throughout the body. These organs can be classified on the basis of their functional differences as primary lymphoid organs or secondary lymphoid organs. The primary lymphoid organs provide appropriate microenvironment for lymphocytes development and maturation. However, the secondary organs to trap antigen from the defined tissue or spaces and provide sites where mature lymphocytes and antigen presenting cell interact with each other to initiate a specific immune response. The circulatory system and the lymphatic system interconnect these organs, uniting them into a functional whole. A variety of white blood cells or leukocytes participate in the development of immune response. We broadly classify the cells of immune system into cells of innate immunity and cells of adaptive immunity. The cells of innate immunity include macrophages, dendritic cells, mast cells, natural killer cells, granulocytes. And the cells of adaptive immunity include T cells and B cells, which are called lymphocytes. So these lymphocytes are the only cells which have the attributes of diversity, specificity, memory as a hallmark of immune response. So you can see there are few cells which are involved in both innate response and adaptive response, which include gamma delta T cells and natural killer cells, T killer cells. So let's discuss them in detail. All the cells of immune systems are derived from a common progenitor cell present in the bone marrow. This cell is called pluripotent hematopoietic stem cell. Pluripotent means the cell has the ability to differentiate along a number of different pathways, and it can generate a number of different types of cells. And the term hematopoietic is derived from hematopoiesis, that is the formation and development of red and white blood cells from the stem cells. So this cell has the ability to develop and differentiate into different types of red blood cells and white blood cells. The progenitor stem cell can differentiate along any of these three lines of differentiation. The first lineage is called erythroid lineage, in which the stem cell differentiates into erythrocytes, that's the red blood cells, and megakaryocytes, which develops into platelets. The second type of lineage is called myeloid lineage, in which the granulocytes and agranulocytes are produced. And the third lineage is called lymphoid lineage, in which small and large lymphocytes are developed. As you can see, the hematopoietic stem cell in the bone marrow, when it follows the erythroid lineage, it can develop into an erythroblast, which forms erythrocytes of red blood cells. When it forms a megakaryocytes, it develops into platelets. The second lineage is myeloid lineage, in which the myeloid progenitor is developed. Remember, a progenitor is the one which is developed from the stem cell, but it lacks the ability to self-regenerate itself. So it is committed to this lineage. So it can myeloid progenitor cannot develop into erythroid progenitor or vice versa. So, this myeloid progenitor can result in development of granulocytes like neutrophil, eosinophil, basophil, or agranulocytes like dendritic cell, mast cells, and macrophages. So third lineage in which a lymphoid progenitor is developed, which is committed to development of lymphocytes, which can be B cells or T cells. And these B cells, as we have already discussed, develop into plasma cells, which secrete the antibodies. And T cells are of different types like T cytotoxic, T helper, T regulatory, and this progenitor also has the ability to develop into natural killer cells. Now let's discuss the function and the structure of each of these cells involved in immune system. As the cells which originate from myeloid lineage and the lymphoid lineage are involved in immune response, so we will discuss only these cells originating from these lineages only. So myeloid lineage results in production of granulocytes and agranulocytes. These granulocytes are also called polymorphonuclear leukocytes. That means they have nuclei which can have different lobes, like that's why they are called multi-lobed. So the lobes may be range from 2 to 5 depending upon the type of granulocytes. Granulocytes are generally the first line of defense against an immune response. The second important thing is these granulocytes have large granules in their cytoplasm. These granules store different chemicals or enzymes within them. So there are different types of granulocytes. Let's discuss them one by one. The first type we're going to discuss are neutrophils. These neutrophils have granules within the cytoplasm which are stained by both acidic and basic dye. That's why they are named neutrophils. The nucleus has lobes which may be 3 to 5, and these lobes are connected by threads. The granules in the cytoplasm are stained generally light blue in color. So neutrophils are the major types of white blood cells, that is they range from 50 to 70%. These neutrophils have phagocytic activity. That means they can intake the bacteria or microbe by means of endocytosis and digest it. So these neutrophils have those granules which are classified as primary or secondary granules. The primary granules have peroxidases, lysozyme, or other hydrolytic enzyme in them. The secondary granules are relatively smaller in size and store collagenase and lactoferrin. Together, these granules helps in digestion of the endocytosed microbe. The second type of granulocytes we are going to discuss are basophil. They are named basophil because the granules and the cytoplasm are stained with a basic dye, generally methylene blue. So these granules are generally blackish or bluish black in color. The nucleus is two-lobed and they contribute about 1% or less than 1% of the total white blood cells or leukocytes of the blood. These cells are not phagocytotic, but they have the ability to change the permeability of the blood vessels, that is, they are vasoactive mediators. Their granules contain different chemicals like histamine, prostaglandin, leukotriene, serotonin, and they have essential role to play in allergic responses where they release these granules, and they are also involved in response to a parasitic infection. The third type of granules we're discussing are eosinophils. They are named eosinophils because the granules in the cytoplasm react with the acidic dye, that is eosinophil, and their nucleus is two-lobed and which is connected by a thread, and the granules are generally stained red in color. And these are mobile and able to move like the neutrophils, and are considered the primary defender against parasites, and they also are phagocytic. But these eosinophils generally contribute to 1 to 3% of the total WBCs in the blood. The fourth type of granulocytes are called mast cells, which are generally present in the mucosal membrane, connective tissue, and have role in inflammation and allergic response. They have granules which stores histamine and are also vasodilators. So students, it's time for our first concept checkpoint. Try to answer this question, and there are four different options. We'll discuss their answer in our interactive session.